Welcome, everybody. This is our third lecture in the series, and I'm delighted to welcome Professor William Ayliffe, who is coming to talk about the evolution of eyes to us today. And William works at the Lister Hospital in London as a consultant ophthalmologist, and he is a professor at Gresham College and a professor of a very interesting way, a professor of physics, which is a term that is always interesting, medical and biological sciences, is my understanding. And he did his PhD in corneal transplants and rejection, essentially an immunology project. Now, William is not talking about that today. He's going to inform us all about the evolution of eyes. William, welcome.
Thank you. Thank you very much. Thank you very much for that kind introduction. Thank you for asking me to come, and thank you all for attending on a wet and an interesting winter night. Eyes are just amazing, and any of you who are not in the optometry department, you still have time to change your courses from whatever you're doing and go and do about eyes, because you will never, ever regret it. This image here is of an arthropod, so imagine something like a lobster. And like a lobster, it has a compound eye, as you can see, with many different facets on it. This particular arthropod died 550 million years ago during the Cambrian period. And we know a lot about its lifestyle. It would live buried under the sand with just these eyes sticking above, where they could look and survey the world in 360 degrees and find their prey, and also protect them from predators that would like to eat them. This is made of calcium. It's hard. It's a crystal structure, and it can't be damaged. This eye appeared from nowhere in the blink of the geological record. And what I want to do today is to try and give you a sense of awe that gave me when I first looked at this fossil and fossils like it. Now, there are many, many types of eyes. And eyes of one kind or another are present in nearly all animal species. Why? It's because vision is terribly important. Your body is just a life support machine for the eyes. And once you realise that, you then move out of things like transplantation and organ biology and medicine and so on. You go into ophthalmology before it's too late. And once you do that, you then begin to understand what a really good choice that was. Now, because eyes are important, we'll look at some designs of eyes. All of these are nice little camera eyes. And you might think that they're all related. Well, they are. They've got pupils and they've got lenses, and these can constrict these irises, and some of them have corneas and some of them don't. But essentially, it's the same design. And you might think, aha, they're related. And this is interesting, you see, because we could look at these animals here, and they all look like a fish. And you might think they're related. Well, they're not. This is a lizard, this is a mammal, this is a fish, and they're completely unrelated things, but they look the same. And this is called convergence. And in evolution, what we tend to find is that if you need to swim through sticky fluid like the ocean, you want a shape that's long and thin, and you want some stabilizing things on it. So you develop fins, or you make your arms and your feet into flippers. And you do that later on when you come back into the sea if you're a mammal. And interestingly, you do exactly the same thing if you've got an eye. This eye belongs to a mollusk. This eye belongs to a jellyfish. This eye belongs to one of my colleagues. This eye belongs to a fish. So we have things here that look the same, but clearly are not the same, and clearly have evolved from completely different points. So, looking at this array, it's very difficult. Even Darwin had difficulties with this. So, if we see something so complex and so beautiful as this piece of kit here, is it possible to imagine this could have evolved through a series of small, gradual steps from a single cell? Or is it more likely that this was designed? And at the moment, you may know there is a heated debate about whether the human eye was possible to have evolved, or whether it was actually designed by a designer or a God. This argument is not new. In fact, it goes back to the ancient Greeks. But William Paley sort of crystallised it as the watchmaker argument. He was a very bright chap, the senior wrangler at Christ's College in Cambridge. He was a supporter of the American colonists, so he wasn't daft there, but he was anti-slavery, and those two points were sort of in conflict and still weren't resolved for over 100 years after this. He wrote a book on the principles of moral and political philosophy. Now, the problem was that he said if you were walking across a moor and you turned your foot on a stone, you wouldn't think twice about it, it's just a stone. However, if you walked across the moor and you turned your foot and you found that there was a lovely pocket watch there, you might think that that hadn't just formed out of the soil and might have been made by a matchmaker, or a watchmaker, sorry. So you have a divine person who would make something that's complex. So complex things cannot just occur out of the blue. Now, funnily enough, Darwin had to read his books when he was a student and read sort of evidences of Christianity. What Paley was doing here was to say that because nature is complex, we therefore have a God. And he used the complexity of nature as a moral argument, a philosophical argument to support his views on religion. He was controversial, and he used a parable of the pigeons which criticised the system of property ownership and the bloody code, which led to him not being appointed to be a bishop. And the king said, "Bloody pigeon Paley, we're not having him as a bishop." And that was the end of his career. So be careful on who you talk to if you're a young scientist and you're trying to come along with any views, even views as simple as pigeons. Pigeons are very interesting. We'll be coming on to them later, hopefully. Now, there are many sources for this. You can come back and look at these online if you want to. There's a couple I'll point you to. Most of the pictures, the drawings, are my own or my own photographs. If they're not, they're from Wikipedia. And if they're not from Wikipedia, you'll see a little name on the bottom from whom I've taken them from with their permission. A super book here on Animal Eyes by Michael Land and Derek. And any paper they have written is worth reading. My father, who introduced me as he was an evolutionary biologist, particularly with plants, and introduced me from an early age, and we'd go to all sorts of places digging up things, gave me a lifelong interest in this subject. And my friend and colleague Marcel, who is a geologist, who taught me what I needed to know of the geology to hopefully try and explain to you where some of this information comes from. Now, there was many early theories of natural selection. Thomas Malthus, of course, wrote the famous one on the essay of Principles of Population, and you'll hear this, it's currently in debate at the moment, Malthusian, essentially that if populations become too big, they outrun their food source and then they die off. William Wells was very interesting. He was born as a colonial in America, came to this country, and wrote an essay which was, there's a difference between white people and black people, the white and the Negro races, because the Negroes can survive in climates where white people can't survive so well. And maybe this was because they've been selected over many, many generations. We now know this to be true, and this is a very good example of natural selection. Now, James Pritchard, he'd thought that there were various causes that acted on the single human being, Adam, which had made Adam to appear phenotypically different and permanently different into different varieties or races. And Sir William Lawrence, who is a Bart surgeon just down the road here and an ophthalmologist, similarly the same as me. I'm a Bart surgeon and an ophthalmologist. I'm not a medical reformer. I seem to have failed to manage to achieve that with the current changes that are going on in the NHS. But nevertheless, in 1819, he was forced to withdraw a book after fierce criticism on blasphemy. And in those days, blasphemy was actually tried in this court, which is the Court of Chancery. And this was very serious and totally destroyed his career. However, Darwin, 60 years later, recognises him and says Wells distinctly recognises the principle of natural selection, and this is the first recognition that has been indicated. Now, in other fields, people were looking at natural selection. Now, artificial selection was that you have a brown chicken and you have a black chicken and you mate them and you end up with brown and black chickens. But the idea is you want to end up just with black chickens. So you take the blackest of the brown progeny and you remate them, and eventually you'll end up with black chickens. And that's artificial selection. And that was well known, particularly at this period, because of the increase in population of the industrial era. Britain had to totally modernise agriculture and became very successful at it. Most animal species that are throughout the world that are used derive from the experiments done in this period of English history. Also improving crop yields and using fertilisers. Again, very, very good experiments and really important and driven by the need to feed the population. On naval timber, Patrick Matthew. He works out that maybe if we selected trees that look like this and then we chop this off and eventually we could just grow trees that look like ships, and then we can cut out the middleman and just have forests of trees that... Now, unfortunately, Darwin hadn't come across this, and Patrick Matthew wrote to him from Scotland, complaining, saying, "You haven't recognised my work, you young whippersnapper, and you didn't put it in your book on natural selection." So Darwin writes back to the Gardener's Chronicle, saying, "I've been much interested by Mr. Patrick Matthew's communication, a number of your paper, blah, blah, blah. I freely acknowledge that he has anticipated by many years the explanation that I've offered for the origin of species under the name of natural selection, but I think no one will feel surprised that neither I nor apparently any other naturalist had ever heard of Mr. Matthew's views, considering how briefly they're given and that they appeared in an appendix to the work on naval timber and arboriculture. I can do no more than offer my apologies to Mr. Matthew for my entire ignorance of his publication." Meow. Now, Charles, of course, had developed this fantastic theory, but he took a long time going about it. He studies medicine in Edinburgh. Interestingly, his uncle had also studied medicine, but had died as a medical student cutting his finger in the anatomy theatre and died of septicemia within 6 hours. And that was not an uncommon thing to happen in those days. Charles himself, who was named after the Charles Darwin who had died, hated medicine and wanted to leave. He left to read theology at Christ's College and, of course, was influenced by Paley, Pigeon Paley, or the watchmaker. Now, he becomes a gentleman scientist from HMS Beagle, pictured here, beautiful ship, and goes off round to the Galapagos Islands, where he notices that the same animals look different on different islands. And what he does is propose that there's a tree that happens where we have a prototype. It then branches, and this prototype itself can branch, each one becoming more and more highly selective to what the food is on that particular island that may be different to the food or the other environmental factors that might be present. So the serial, gradual gradations in complexity. And then he says, well, if that can happen to finches, maybe that could happen all the way back. And maybe all organic beings, that's all animals and plants that have ever lived on this earth, descended from some primordial form into which life was first breathed. So we'll trace this tree back. This is us. We come back, more branches, and then eventually we come to the primordial life. Meanwhile, while Darwin was collecting his species and living down in Croydon in Down House, which is well worth a visit for those of you who ever happen to pop down to Croydon, Alfred Russel Wallace, who'd been a teacher in Leicester, gets completely bored. By the way, he was born in Monmouthshire, but hardly spent any time there, so the Welsh saying that he's Welsh is become quite an issue recently. He was born in Wales, but as the Duke of Wellington said, and he was born in Dublin, he said just because one is born in a stable does not mean that one is a horse. But he collects the beetles with the naturalist Henry Bates, and he read the account of the voyage of the Beagle to South America, and he goes off himself, and he collects a ton of stuff. But unfortunately, on the way back, the boat burns in the mid-ocean. They've all got to go off, and they're in open boats, where luckily they're rescued by a passing ship in the mid-Atlantic. So he comes back to England. And after a few years, he gets someone. So this time he goes off the other way around the world to Malay and collects 125,660 specimens, including 1,000 new species, to science. It's astonishing work. He worked out that the species are different, separated by this imaginary line that still today is called Wallace's line, where the two distinct faunas, the Australasian faunas and the Asian faunas, are separated. And then he realizes that there's something to do with the most appropriate progeny survive better than the progeny who are less adapted to live in that environment, and writes to Darwin. Darwin has a problem, because this is now ready for publication, and Darwin had thought about this decades earlier. So he seeks advice from Lyell, and Lyell says, you have a problem here, but what we'll do to solve this is we will read both papers together simultaneously at the Linnaean Society, which they do, and then Darwin subsequently goes on to publish The Origin of the Species presto pronto. Now, Wallace was never bitter about this and always recognised that Darwin had anticipated him and didn't claim that he had published first. And his claim to fame, he said, was that I encouraged Darwin to publish without further delay. Now, Darwin, of course, was a figure of ridicule, and here's Darwin and Wallace in the zoo, one clapping the other. But Darwin had a problem with evolution, and it was Paley. It was, how could something as complex as the human eye have evolved? To suppose the eye with all its contrivances, adjust to different focuses, it amounts of light, it corrects different things. It seems to be, absurd to the highest degree. And he writes to Asa Gray about this. What the creationists fail to mention next is the next paragraph, which says, but nevertheless, there must be bits missing in the fossil record, to paraphrase him, that actually point out that this could happen by small incremental changes. Richard Dawkins wrote a very influential book and series of papers called The Blind Watchmaker, and not being afraid of controversy in any way, promotes evolution, if you like, in a very avant-garde fashion, and is very anti-religion. Now, the story is that if you have a monkey and you give it long enough, it could eventually write Shakespeare on a typewriter, which would be difficult because the last typewriter was made in North Wales 4 months ago and they've closed the factory. But nevertheless, assuming that we could get a typewriter and it wasn't going to wear out. Well, that is true, but let's make it a bit simpler. Let's just give the monkey 28 different characters and get it to make a sentence. And the possible combinations are then 27 to the 28, which is about 10 to the 40. So if he's got a single-step selection, it would be about 1 million years to type out a single sentence of Shakespeare, never mind the whole works of Shakespeare. And that's more than 1 million times longer than the universe has existed. However, what you can do is if you make a random error, and that error is very much like where you want to go, you can speed up this process. And this is exactly what Dawkins did with this computer program. Here is the sentence, methinks it's like a weasel. It comes from this part of Hamlet, where Hamlet is feigning madness, and his mother wants to see him, and the actors come in front of him, produce the play, and then Polonius comes up, my lord, the queen needs to speak with you, and presently. And then he says, do you see yonder cloud? It looks like a camel. By the mass, 'tis a camel indeed. They're trying to humour him. And he says, no, methinks it's like a weasel. And on it goes. So, methinks it's like a weasel. Generation 1, random series of 28 letters and spaces. Generation 2, S, is actually correct. So we're going to keep that. By generation 10, we've now kept this, starting to look reasonably similar to where we're going. By generation 40, generation 43, methinks it's like a weasel. So instead of 1,000 million years, this has taken a short lunchtime on a computer. Now, you can use this as an analogy of evolution. You can actually say that if this is fitter, i.e. it's more fitting this, and this is even more fitting than this, and this is even more fitting, we will eventually, in the blink of an eye, get to where we want to go. And that's sort of a fallacy of how evolution works, because evolution doesn't work like that. Evolution, because then we're back to religion again, aren't we? Evolution doesn't say, here is a human being and he's like God. Here is a earthworm in the primeval soup. We want to get from earthworm to human. How do we do this in individual different stages? Evolution doesn't work like this. And one of the reasons it doesn't work like this is because of mass extinctions, more of later. But assuming we're going to do a computer program like this, and we want to get to a human eye that's like a camera eye, what we're going to do is to start off with a skin, and some of that skin has photoreception, which isn't uncommon. In fact, it's very, very common. And here's the photoreceptor here. And then eventually we go to another stage, through a series of stages, where we shield that photoreceptor with a little bit of pigment. And this means we've got some direction. It means the transparent animal now doesn't see light from below, it only sees light from above. And it multiplies the photoreceptors. So we've got a little patch of photoreceptors on the surface. And this, again, is very common in biology. However, it's not very directional. So what we want to do is to curve it even more. And eventually we end up with this, which is very directional. This thing can actually form a simple image. And there's lots of these examples in the biology world. So this is getting very close to an eye. But the problem is it's non-focused. The focal length is down here. But if we then, instead of having seawater flushing in and out of here, fill it with some sort of gel, we now get a weak lens. And this weak lens starts to focus about here. And what's interesting about focusing about here is that we're now only stimulating 1 or 2 photoreceptors, which means that we're getting a much sharper image, and we can see different things in our environment. Then we condense the lens, rather like fishes did, and now we've got a gradiated lens, which can really start focusing things pretty close to where we want them to be, and get reasonably focused images. We can then shield the whole thing with a cornea, and then we can start separating them up and getting really good focused images. So you might think that would take a long time. If we were parsimonious about the change and say there was a 0.005% change in each generation, that would take 364,000 generations, which if you're dealing with animals that have 1 year, that's a blink of geological time. You cannot detect that in the geological record. You can't detect it in the geological record if you've actually got progeny of 10 years or even 100 years. So it's very difficult, and that's why you can appear after a major extinction event in the Precambrian and suddenly in the Cambrian appear with that eye that I showed you, as if from nowhere. It's not from nowhere, because the period of time in between that is millions of years, which, as we've just shown you here, is plenty of time for a gradual evolving to do that, if that's what you wanted to do, or if that's what nature wanted to do. And I'm going to come back that although this model is very plausible and shows us how we might have evolved an eye, it's not necessarily how an earthworm or any other animal might have lost an eye, or gained an eye, or had an eye that was of a particular type. But if we are going to make eyes that are complex, either having compound eyes or simple eyes, or I prefer the term single-chambered eyes because there's nothing simple about this at all, what we have is a number of different steps. So we have a non-directional response. Now we want to have direction. That means we want to know where the light's coming from. So we need to shield it. And we can either shield it by putting pigment spots in here, rather like cnidarians do. So jellyfish that are going to make eyes have this system. We've also got to pack in the photoreceptor molecules, millions of them, and we've got to pack them in very tight. So we've got to fold up the membrane. And we can either do that by growing out a hair and making branches, so-called cilia, or we can do it by making little villus extensions, like in our gut, and they're called rhabdomers. So we have ciliated and rhabdomeric receptors, and then we can not have pigment actually in it ourselves. We can have a separate pigment layer, separate pigment cell, and we've got very good directional responses. This one's good because not only is it not from below, but equally you've got to be pretty accurate. It's got to come really from dead on top. This one just shields it from below, but anything from the sides or whatever can get into it. And that means its spatial acuity is reduced. The next thing now, we want to improve spatial acuity, so we make pinhole and cup eyes, and then we add in a lens, and then we're really up and going. Now, could something like this happen? Well, the answer is yes, because we do have some of these species. We know, for example, that many eukaryotes shield their light-receptive organelles. So eukaryotes are single-cell animals with beings with a nucleus. There's this interesting thing, and this is a real fossil. What this has is one cell here, which is pigment, and one cell here, which is a nerve, And that nerve comes down, and when it fires, when it sees the light, it contracts this band, which then starts these beating. So if it sees the light coming from this side, it beats those, and the animal turns, and it turns towards the light. You could have it the other way around and have an animal that turned away from the light if you wanted to, and we're going to see some examples of that later. But this is very basic. There is one neuron, one pigment cell, very much like I showed you on the last diagram. And what's amazing is it exists. So the theory is here. Maybe we are talking what Darwin's talking. Maybe it is possible to evolve by small incremental steps this wonderfully complex piece of kit, which is the reason why we're all here and seeing what we're doing here, called the human eye. Well, the most important thing we've got to develop first, if we're going to go down there, is the chemical machinery to do it. And this is a very particular molecule that we are going to evolve in this story a couple of times, but only a couple of times. And once we have evolved it, we don't lose it. We keep it for billions of years. We now pack in the photoreceptor here into the membranes. We then add in a few other things. We add in some clear cells between. We add in some neuron connections. And then eventually we package it up. And there's only a certain number of ways we can package this up. And this is them. We can have a pinhole eye, and we can have multiple pinhole eyes. So everything here is single-chambered eyes, and everything down here are compound eyes. We can have lenses, focusing systems on them, and there's two sorts. There's appositional, where they're opposed, and there's superpositional, where they're not opposed, and there's a little clear space in between. And these give different types of images. This gives an erect image, unlike this system, which gives an inverted image, and a number of other issues here which are quite interesting about seeing in the night time. These give multiple different inverted images, which are then combined by the neural system into one image. We've got reflecting systems that actually comes in, it hits a mirror, and you get parabolic reflectors. You also get different reflecting systems in compound eyes, where they can be reflected down the shafts of transparent crystals. So, looking at all these eyes, von Salvini, Plowin and Mayer decided they'd agree with Darwin. Eyes must have arisen independently many, many times. However, when they were messing about with fruit fly transcription factors, which if you're messing about with fruit flies must be one of the best things you can do with them, I would imagine, they found a protein that bound to DNA. Now, proteins that bind to DNA are a feature of transcription factors, and when you find them, they're quite important. So what you immediately do is you run to the computer, and you look on the computer and see, has anyone found it before? No. You then run to the other computer program, and they say, does this look like anything anyone's found before? And amazingly, it did. Now, what they found was that this protein was homologous to the mouse small eye PAX6 gene, the human aniridia gene, and a bunch of other genes that are all associated with things to do with the eyes, either assembling eyes in the fetus or with eye diseases in humans. And they suddenly realized that, in fact, all animals, all metazoans, that's multicellular animals, have the same toolkit. And it's related. It's a very... jellyfish even have something related to this. Never use it, except in two phylum, where they do use it. And what's interesting when they do use it, they make an eye that looks very much like ours. The other jellyfish don't. They just sort of wander about sort of seamlessly on the ocean currents to and fro. The ones that have eyes can see and can chase predators, and they're deadly poisonous. Now, so one toolkit allows animals to develop eyes de novo when they need it. But what about the chemical toolkit? Well, that is sort of rhodopsin and opsins. Now, these are proteins that bind a pigment that can see light, or detect light, or absorb light would be the best word. It doesn't actually see yet. We haven't invented the eye. So we're absorbing light, and then we make a conformational change in that protein that does something to the cell that contains it. It might give it a second messenger. It might do something else in flies, and we'll talk about that later. We have single-celled things called bacteria, and we have other single-celled things called archaeons. These still exist. These exist in our guts. They're the reasons why, that after a couple of pints of beer, you have a rumbling in your tummy. They're called methogens. And eukaryotes, which are single cells again, but, well, actually, and us, but protists are the single-cell versions of eukaryotes. These are cells with nucleuses. And you can see that our version of how this all develops is similar to Darwin's tree as well, as you can see from the page in his notebook. Now, these prokaryotic single-celled animals with the nucleus evolve an opsin. And, funnily enough, it looks very much like our opsin. And originally it was thought that this opsin must have then been carried on down through evolution. Unfortunately not, because we have a different type of opsin called type 2 opsin. And our opsin, uniquely, is linked to a very special protein called G protein. And if you link receptors to G proteins, you can do enormously fantastic things with it. Nevertheless, we did descend from these animals, so let's see how things might have happened. These animals, by autophagy, might have eaten and absorbed each other and collect together a bunch of stuff within them. For example, our mitochondria may well have come from an ancestral bacterium that was engulfed. And there are other things, such as chloroplasts in these animals, may well be a chlorophyll bacteria that was engulfed, and now they live in symbiosis. Then, eventually, there's intermediate stages, and this exists, it's called Volvax. We eventually develop a metazoan, meaning multi-cells, and some of these go off and don't differentiate much and come into sponges that have no true tissues. Some of them develop radial symmetry and go on and form corals and jellyfishes. Some develop bilateral symmetry and 3 germ layers, and form this prototypical bilateral animal. You can see it can be divided into 2. And some of these develop no gut at all, just have a hole in the middle, and flatworms would be an example. Some of them develop a pseudo-coelom around the gut, and these are roundworms. Some of them develop a complex space here, which can then divide into 2. It depends whether you develop the mouth or the anus first. And depending which way you do that means you become a protostome, where you can become things like mollusks, or you can develop segments, and that means you can have things like a head and a tail and arms and legs. Imagine a lobster, if you like. Or you can become a deuterostome. And the thing about deuterostomes is they can come on and develop things like backbones and central nervous cords, and they become things like chordates. And interestingly, starfish are also deuterosomic as well. Now, that simple tree has become slightly more complex because looking at analysis of mitochondrial DNA, and we now know that essentially it was right. The parazoa that have no tissue is here, radial symmetry with Cnidaria and Stenophora. But the difficulty comes here is when we divide the protostomes, and we have these Lophotrochozoas, which have ciliated tentacles around the mouth. More of that later, because many of these have eyes, and the ones that molt, which become nematodes and arthropods. And then our tree, the deuterosomes, that comes on and eventually comes down through chordata and has a bunch of really interesting animals and develops opsins, and we'll talk about those later. And this weird thing that lives in the bottom of the silt in a Norwegian field that turns out to be closer related to us than any of these other things, which was a bit of a shock to biologists when they found that. What is interesting is look what's happening here. The villus type of photoreceptor is going to go in the retinas of most protozomes, and the ciliated one is going to go into the retinas of the deuterostomes. Pretty much entirely. There are many exceptions to them, which I will come to, but those exceptions don't make the rule. So if we imagine this here, this is Bart Simpson with a short haircut, okay? And then this is Marge Simpson with a tall haircut, okay? So when I come on to that, if you remember that later in the lecture, we'll find out where we are. Very quickly, for those who are not eye biologists here, which is probably about 2 of you in the room, what was going to point out about these receptors is that light hits them, changes the protein then becomes energized, it then sets off a cascade and releases some second messenger, which then can open channels and allow ions to come into the cell and can change the polarity of that cell. So it can hyperpolarize, or it can lose ions and become depolarized. And it depends on what sort of animal you are, which of those you do. And it does it very, very quickly because of G proteins, okay? Even jellyfish have G proteins, not quite the same as ours, but they use this as well, because it's got to be quick. Now, even more interestingly, in things like flies, it's even quicker. And what evolved here were two types of these photoreceptors, I've said. So we've got Marge Simpson here, so I coloured that helpfully in blue for you because she always has blue hair. And this system uses a second messenger, and it's rather slow. But what's interesting is that this is active in the dark, okay? And it's in the light, it's turned off. And that's really fascinating. That's us. Our retina, if you shine a light in our retina or like this, it doesn't actually do very much. It turns on if you put something dark in it. And this was really interesting when they did the very, very first experiments on vision. The opposite is true of the rhabdomeric. These things respond to the light, and they respond very, very quickly. They have a phospholipase C here, which as soon as they get the conformational change here, lyses the membrane, which then changes shape. And what's interesting here is they don't need a second messenger. This change of shape pulls open those channels I told you about. So it's instantaneous. This is mechanical, not chemical. So everything I learnt in medical school was wrong. Okay, this is a mechanical opening. And mechanical receptors, the quality they can do very, very fast. And that's one of the characteristics of insect vision, for example, that uses this system, is they're very, very fast responders. Now, we mentioned that different types of receptors tend to go with different types of animals. So we're getting the squid, Drosophila, they're all inheriting the blue type of way of packaging, whereas chordates, we're tending to inherit solely just the red type, or the ciliated. Now, the problem with this is that it's all wrong, because we've suddenly found in the human retina that we actually have insect-type receptors. We don't use them for seeing. We use them for circadian rhythm, for setting the time in day and night. We also use them, I think, for giving us migraine. And what's also interesting is that I think that we can see polarized light with them. And that's why we can see Hadean brushes. And that's why animals that have ciliated receptors normally can't see that. But if in your retina, like humans do, you have something that belongs in an insect retina or in another animal's brain, for setting circadian rhythm in that case, you will see polarized light. And I'll come on and show you why I think that in a case. So in summary, just very quickly before we move on to some real animals and some real evolution and real biology, we'll just go through very briefly. We know ancestral genes were here. We know that this animal must have had both types of photoreceptors because we find them in all these animals down here. We know that the gene for opsin, the one that actually changes with light, is highly conserved and looks very, very, very similar across all different types of animals, and we know it comes in two types. One type is used in arthropods and insects to see with, and they use the other type in their brains, and the backboned animals use it the other way round. We have the insect type in our brains for setting circadian rhythm, and we use the other type ciliated for seeing. Now, I don't have to tell you that the Earth is very, 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 very old. In fact, it's so old that if 1 year was 1 millimetre, this line would be 4,600 kilometres long. And dinosaurs appear for the last 160 kilometres and then become extinct, and humans actually take this last tiny dot there, which is an over-exaggeration due to the limits of this lecture theatre and slide not being big enough. The Earth is divided into the Hadean Period, the Archean, the Proterozoic and the Phanerozoic eras. And just some words that will come out, Cambrian, Ordovician, you'll have heard of all of these. Ediacaran you may have done, and I'll talk about them. Now, realising the Earth was very, very old, and that there was a reason that there were many species there, actually comes about really quite late on. Nicholas Sterner was the first to perhaps think that when he was walking up on the mountains in Italy and collected some interesting bones, he was then asked by the Pope to, interesting stones. He then dissected this shark, which had been swept up on the beach in Livorno, and then found out his moonstones or magic stones were identical to the teeth in the shark, which meant that one of two things. Either sharks used to live on land and lived on the top of mountains, or that the top of mountains was once under the sea, which meant that the Earth wasn't static and changing and it was moving. So why were there so many animals out there? Well, the original view was that animals were out there as God's way of keeping meat fresh until we invented the refrigerator. Now, more sense, and then he gives up after this. He goes into religious orders and stops writing scientific papers. James Hutton comes along and intriguingly works out a lot of very interesting stuff. He realizes that the processes that go on now were going on forever, for millions and millions of years. He also worked out, looking at this is called a nonconformity or an unconformity. These must be older, these are newer, and because they're settled flat, something happened to these that pushed them up in olden times. They then went down under the sea and new layers were deposited on them. So the Earth is undergoing cycles which must be taking unimaginably long times to do this. And then faults or pushed up, they're pushed up above again, they then erode, and the process continues. And the silts and sands from those go in and get deposited out on their daughter rocks, which are being formed here and further out, and further and deeper down. And you can use these strata to date things, because William Strata Smith realised that if you were looking in one strata in Devon and you looked at that same strata in Yorkshire, they actually had the same fossils in them. And you could actually date the strata. And he made this wonderful collection and built this museum, which still exists today. Unfortunately, the contents don't, where he displayed them. And you walked in and you walked around this and you walked through geological time. It was quite amazing. And he actually dated the Earth. And it was really important. This is his map. Now, this is where these terms come in, because they're a bit confusing. So Cambrian was the earliest name for Wales, because that's where you find Cambrian rocks in the olden days. And Ordovician and Silurian were the names of the Welsh tribes, which is essentially still there today. You've got the northern Welsh and the southern Welsh, the Ordovician and the Silurians. You've got Devonian, one speaks Welsh and one doesn't. Then you've got Devonian, which is named obviously after Devon. Carboniferous, which was an adaptation of meaning the coal measures. Permian, now that's not English, that comes from a strata in Perm in Russia, but was found by a British geologist, Roderick Murchison. Triassic, this is purely non-English, this is German. And here's an example of it. And you can see it's 3 different layers, white, brown, and orange. That's why it's called Triassic. And these are the Triassic beds. And then Jurassic was named after the Jura Mountains. So when you see these names, don't let them confuse you. There's a reason why they're there. And once you know why they're there, they're actually quite simple and they're quite fun to remember. Not much geology during this period, because this is when the Earth was molten and various things were happening and planets were bashing in and the moon's going to form. But within a very short time, geologically, things do start to happen. Liquid water appears on Earth. And we know these things are stromatolites. And what these are colonies of bacteria, single-celled animals. And we can actually go back in time and we can find them in the rocks. And these are billions of years old, and they're identical to these. So early life looked something like this. These, around a very hot spring, are algal mats, if you like, or simple-celled mats of living tissue. This is a road, this is a bus, and that's a car, to put it in perspective. So if you want to go back to the Hadean period, that's what the Earth would have looked like. Okay? They were single-celled, as we mentioned, but what's interesting about them is they used chemicals to harvest light, even at this stage. And they used them very cleverly. They used a rhodopsin to actually drive a propeller to drive it up to the light, and when it got too much light, they used another propeller that turned it the other way around that drove it down out of the light. So it would have just enough light to come up and down. Look at this horrible polluted sky here. There is no oxygen in this. So you've got to use something that's good for energy. And sunlight's good for energy if you don't have much oxygen around. Then oxygen does form, because the byproduct of all of this messing about in the water is oxygen. And eventually it rusts the ferrous iron in the plants. And that's why things like sandstones in the Jura Mountains are red. And that's why this is red as it gets deposited. And it's essentially rust. And the problem with this is that oxygen is very, very toxic. It's toxic to us, and we have mechanisms for dealing with it. It's terribly toxic to the animals that made it, and it wipes them out. There's a mass extinction, and all these little critters all die out, except under certain particular areas, and that's why we call them extremophiles, because they live in extreme environments. Actually, for them, it's home and cuddly. It's what the Earth used to look like before we had oxygen on it. We've mentioned Lynn Margulis, who was considered mad. Probably was mad, but her theory turns out to be correct, as often happens with people who are eccentric. Unfortunately, she's died recently, but very, very influential, reinvigorated Merechowski's system, which is we accumulate organelles by eating other organisms and not killing them. And we make these complex single-cell life. Now, we tend not to be very much interested in single-cell life because they tend to be things that are horrible, like slime nets, diatoms, algae, muddermilk, and they tend to cause diseases like the Irish potato famine or things in your gut. So they're highly unpleasant things. And they're also unpleasant because we look at them when we do O-level biology. I didn't do O-level biology, unfortunately. But, this is euglena, and it's got a little eye spot, and this eye spot's attached to that flagellum. Hey, we've seen this before in animals a billion years earlier, haven't we, which are used to turn and drive it using light. And here's another animal with a little flagellum. We now associate this element with red tides and toxicity. But some of them are very complex. This is one cell. And look, it can focus light onto things that can harvest it. This thing sits at the bottom of the pond. It sees something moving above it, and it leaps up and it eats it. And then it goes back down to the bottom of the pond. Scary. It's only one cell over. Now, eventually this single-cell life becomes multicellular and forms this very interesting biota called the Ediacaran biota, originally found in England but became popular in the 1940s and takes the name from this area in Australia. And multicellular life, very controversial even to this day. Some people think these are all unique and they all died out and they're not related to modern life. Other people think, however, that this impression actually relates to something like this, that's a mollusk, and that is related to things like mussels, and that they came through. Particularly at the end of this period, the calcium was able to form in the seas, and small shelly former arrive. And there's a lot of probables. Maybe all of these developed in this period. However, maybe it didn't. Some of them may have even had legs. Maybe they didn't. It's difficult to interpret the fossil record. But they've all gone. So something happens. And you probably guess what that something was. That was a mass extinction, which wipes out the entirety of the Ediacaran biota, leaving behind an amoeba, which seems to be the only survivor from that period, unless there are missing links that survive that come across, and those things really were early mollusks and early jellyfishes, which is very controversial. This is the guy who named it, the Cambrian era, by the way. And we get an explosion of life, including some of our favourites, the trilobites, and some of our less favourite things. These critters look pretty ugly, and some of them really were ugly and rather nasty predators, including Opabenia. Now, Opabenia, you'll notice, has several eyes. It has 5 eyes. It has 2 on stalk, so it's got bilateral cephalic eyes, we'd call that. And that's really important, because we've got bilateral cephalic eyes, 2 eyes. So do insects. And insects also have a bunch of other eyes. They've also got another 3 sets of eyes in the middle ocelli, dorsal ocelli here. They've also got another 2 behind their other eyes. So having lots of eyes isn't that weird. It's just that the way they do these drawings makes them look weird. They're reasonably large, 70 millimetres, 7 centimetres, the size of a small ruler, if you like. And some of them look truly horrific. They're Anomalocaris, that means weird-looking shrimp. And here it is in the Science Museum with this compound eye, and they've actually found the compound eye down in Australia. So 3 centimetre eyes, enormous, 16,000 lenses, really good. The age of predators has arrived, and they believe the Cambrian explosion, this change and massive amount of life, was partly driven by the evolution of the eye, by the invention of the eye. Once you've invented an eye, and you're a predator, all your prey have to develop eyes, or something pretty smartly, and you've got to do it very quickly, otherwise you don't exist. Now, not only do we have eyes developing, we start developing lenses and complex eyes, and these still exist. These are chitons, and here are eyes of chitons that have crystal lenses inside them. Now, crystal lenses are important because they were also present in trilobites. Trilobites had these wonderful compound eyes, and helpfully I've done a little section. They're all like ice cream cones, and modern animals, arthropods, that have these types of receptors, and you can see how they're aligned in this way that can explain why they see polarized light as well as the normal light that we see. And some of the eyes were terribly complicated, lots and lots of facets. Here's this animal, it's got one on the other side, here's its backbone. And some of them had very complex optics. This had dual optics. This is spherical aberration. If you have a spherical lens, and all these lenses are spherical as you can see, you're not going to get a clear focus because it's going to spread. But if you actually have two different refractive indexes of lenses, you can actually get a sharp focus. And making these aspheric surfaces and duplex lenses was the way trilobites dealt with spherical aberration. Now, unfortunately, this doesn't get rid of chromatic aberration, but that doesn't matter if you're a few feet under the sea, because there isn't any chromatic. It's all blue, because all the red light gets absorbed within a few feet. So I'm now going to move on to vertebrates, because that's the bit that people get really interested in. But I actually like all the other critters we've talked about, like scallops, because they're delicious, and lobsters, and it's really fascinating. And the reason they developed eyes is because they're delicious, and they need to develop eyes to stop predators finding them and eating them. And we'll talk, if you're interested in that, we'll talk more about what those eyes see tomorrow evening, if any of you are around and want to come to the Museum of London, if you haven't had enough of this by then. Well, we're just talking about animals and animal eyes. I'm not going to talk about that now. I'm going to talk about how we develop them. Now, it's another one of these funny trees. Don't let them confuse you. It's just essentially Darwin again. We have a bilateral animal. We come off here, and then we form all of these that we've now dealt with. We're now going to develop chordates. Chordates are animals that have something running down the back, which can go on eventually to form, you're going to get a head and a tail eventually, like in a hagfish, and then you're going to form a backbone, a vertebrate. And that's us, gnathostomes. I gnath my teeth at you. Now, chordates, they've existed for a long, long, long time, and they have a head, and this one particularly had very interesting, conodont had very interesting eyes. We know how they swam because we can see here, we know the fish. And fish, you see, swim like this. When you're a mammal or you're a reptile, you don't have a backbone to do that. You're going to swim like that. So dolphins swim like that. So we know these are fishes and not prototype something else's, because they swim like fishes do. Now, chordates, really interesting. Some of them still exist. We have tunicates, and we have these things, which is Amphioxus, which means the head and the tail look pretty similar. But what's interesting about Amphioxus is it's got a single eye. And this is the prototype of what's going to become our eye. And it's way back in evolution here now. So we're talking 500, 480 million years ago, to put it in perspective. A very, very long time ago. And not only is this single frontal eye similar to ours and has similar anatomy, it uses the same signaling cascade that we do. So we now know, since this evolved from here and shares what we have, this thing, the primordial chordate, must have had the same in its eyes and must have had similar eyes as we do. And let's take hagfish, for example. This is one of the most disgusting creatures God ever invented. It does have an eye, but it's covered by an opaque skin. And it has a rudimentary structure. And it has a bilayered retina. And these ganglion cells project to the hypothalamus. The reason it's disgusting, when fishermen catch this, they throw it overboard. What it does is it goes inside the animal and eats it from the inside. And it does this to animals that lie on the floor. So you don't want to go and sleep on the ocean floor next to these, because they come at you, thousands of them. And there are documented incidences of live tuna being attacked by these things and eaten, eviscerated, just leaving the skin on the outside. Which I suppose, say, spitting it out, considering you haven't evolved a jaw, that would be rather, you'd probably choke, wouldn't you? What you need to do that, to spit things out, is to develop a jaw. And we'll come on to those in a minute, because there's one more interesting animal that doesn't have a jaw that's called a lamprey. This is what I mean, it doesn't have a jaw, it's got that. And that sort of latches on and goes, and it eats its food in that rather pleasant fashion. Come to our mess at some stage when we were medical students, and you'd know exactly what I'm talking about. Now, the interesting thing about this is that it's developed a camera eye. We're developing sophisticated eyes as we're coming down this tree. And they have opsins, and they have long wave, short wave. They've split the opsins into two, and they've even got a rhodopsin as well. And it's got a retina like any other vertebrate. And what's interesting, when it's a larva, an ammonocete, it has an eye that's a little bit like the hagfish eye. And as soon as it matures, it develops an eye that's very, very similar to ours. Now, some people like these animals. Lucius Crassus kept one as a pet, and he loved it so much, he actually put earrings onto it. And he was called a hypocrite by the censor, because he cried more over his lamprey when it died than he did over any of his 3 wives. You may know the story of Henry I as well, who died on the 1st of December. He's fourth son of William the Conqueror, and he died of a surfeit of lampreys. Too many. Paleozoic era is going on. I'm just going to leave this because simultaneous to developing these backbones, we have the non-backboned animals coming on, developing very complex eyes, some of which were the most interesting. This has two retinas. It's got a ciliated retina here, like ours, and it's got a rhabdomeric receptor here, like other arthropods. And look at these beautiful eyes. Can you see them? I don't know if they project, but my goodness, don't you fall in love with that animal. And then the reason it's got those eyes, as I've told you, because what's inside it is really one of the best things that there is on possible to eat with black pudding, which is the muscle that closes the cell shut. It's abductor muscle. It's the only one of the bivalves that can swim, so it needs to have darn good vision to avoid other things that might want to eat it. This is a pinhole eye, not very good optics, and particularly bad optics if it needs to let light in at deep depths. But all it's going to do down there is to see large animals coming at it. Fish start to develop in this next period, 415 sharks, and some really ugly-looking critters with very well-preserved bony bits of their eyes. And interestingly, not only fish, but even birds who descend from these animals still have these types of eyes. The first things on land probably looked like this. They were a myriapod. And these things lived in pools, and they're found in the Rhynie Chert, and these fairy shrimp still exist today. And eventually they evolve into these things that are somewhere between insects and non-insect animals. It's a little bit of taxonomical debate going on these animals where they are. Now, they've come out with very simple eyes, but meanwhile, back in the sea, rather beautiful, complex eyes have developed, and they become very specialized with spherical lenses to see under the sea, because you need a lot of power, because the water on the inside of your eye and the seawater on the outside of your eye have the same refractive index. So you're not going to get much power from a cornea, so you need a very powerful spherical lens. And they develop various other things to do with that, including color vision. And color vision develops really early. That was that critter I showed you that was swimming in the sea all that time ago. We know from descendants that it had at least 4 types of receptors for light. We know that craniates develop a head, split this into 2, and go on to form the jawless vertebrates like lampreys. Down this lineage, we split again, and eventually we form a whole heap of things that can see different spectrum of light and develop sophisticated colour thing in bony fishes, such as the cichlid fishes and other fishes in the lakes in Africa, which are well studied, and zebra fishes. Now, many non-mammal vertebrates also have a green rod, which means they have colour vision in the dark, which is fascinating. We've only got one type of reception rod, so we can't have colour vision because we can't compare it. If you have a green rod and a blue rod, like ours, then you could do colour vision in the dark. That's why all those paintings in the dark are all wrong. But what happens here is that we end up, because of these things, called reptiles, called, let's say, dinosaurs, we as mammals now have to become nocturnal and live in burrows. So we lose all of this. We lose a lot of interesting stuff and end up just with a rod-dominated retina. We've got to re-evolve this, and when we re-evolve it, we don't evolve it as well as reptiles and birds, which still have better colour vision than us, and a whole heap of other things. And finally, the mammals and old world primates split again, and from the long wave we get a medium wave receptor, and this now allows us to compare these two and generate a colour percept of red. And red is really important for ripe fruit in our part of the world, and also for when women are fertile and available for mating, because their bottoms go red if you're a baboon, or their lips go red, and there's no accident that women use red lipstick when they want to appear sexually alluring. So you look at Marilyn Monroe, red lipstick. And this stimulates a very primeval, because remember what I'm talking about here, the whole of this lecture is about sex. It's about natural selection. And it's selection of the fittest. So both the females and the males have to be fit if they want their genes to be passed on. And if you're a female and you are the receptive type of female, there are also donating types of females in other species, you have to make sure you attract the right one. And a good way of doing it is to make sure he's got good colour vision. This was just a slide to show how we can actually fit this in. We now even know the opsins fit into this story. So we're getting information here from geology, we're getting information from chemistry, we're getting from really sophisticated modern studies of DNA and mRNA. There's a lot of oxygen appears around here, much more than we have now, and you get massive insects. Look at the size of this. You really do not want that being a mosquito biting your butt. But luckily for us, there's a massive extinction. We get rid of all these horrible insects. But some reptiles and so on still survive and become these proto-mammals, which I mentioned before. In the sea, you've got these rather large eyes developing. The same reason that giant squid has a large eye is for this. Now, we don't have this anymore, but giant squid still do, and they have sperm whales who eat giant squid deep, deep, deep down. And these are developed to detect bioluminescence, and more of that on another occasion. Birds, we've mentioned, have this very sophisticated colour system. Flies have wonderful speed of reactions, partly due to how quickly their receptors can work. So the simple summary that we've gone before is that there is a very plausible argument that life evolved from very simple things, and we know from genetics and things like mitochondrial DNA that these are very well conserved through time. We have a very plausible model of how eyes might have developed, but not all animals need to develop this eye. They can stop at any level that's good for them. A little annelid worm that lives under the leaf in the Amazon doesn't need to develop an eye like this that's got sophisticated colour vision and all the rest, because if you do that, you've got to develop a brain. And if you develop a brain, you don't have a worm that's this big living under a leaf, you've got a worm that's this big that's living under a tree trunk, which becomes very tasty and edible. So what you could do, if you don't want to have a big brain, you could have lots of eyes. And that's a very good alternative. You can have lots of pinhole eyes. You could do a jellyfish. You could have 4 different types of eyes. They've got 24 eyes of 4 different types. And each of the eyes do different things. One of them looks at the mangroves underneath. One of them looks at the canopy above and directs it to make sure it doesn't go out to sea. And the other 2 actually just act as light and day meters so that they know whether the water's murky or whether the sky is dark and rainy and a storm is coming. So, in summary, I want to say thank you very much. And slightly gone on, but I'm very happy to take any questions. But I would think that from the evidence I've tried to present you there, that although it is quite plausible that God invented every animal instantaneously on Earth and that they're all around now, and that the various complex bits of those animals were given by God at that time, it would leave some difficulties in my mind. One is explaining how you get sharks on the top of mountains that no longer exist. It would also make it difficult for me to put together this evidence that I've placed in front of you, that the massive connections between different complex organisms, they use the same genetic toolbox. If we want to have a simple eye that just detects light, we do that. If we want to detect direction of light, we develop a pit eye. We can do that in the blink of a thing. We can also, as I've shown you, develop a camera eye, sophisticated camera eye, whether single-chambered like ours, or multi-chambered like a fly's. And we can do that in the blink of a geological. So just because Darwin couldn't find the geological bits in between is not an argument against evolution. But there again, nor is everything I've said today an argument against the existence of God necessarily. It's just that I would argue it is more plausible that the eye probably evolved through a series of very small accidental events that led to a small advantage of that particular organism in that particular environment, which is why we can explain the Cambrian explosion, because all the niches were empty, because there's the extinction event, which wiped out every single known species apart from one protist that didn't have an eye. So, and then you develop predation, and then suddenly we're set for the modern scene. So, ladies and gentlemen, I hope that made sense to you, and thank you very much for listening. If you have any questions, I'd be delighted to take them.
Thank you very much, Paul. I not only learned about the evolution of the eye, I did learn a number of other things, that Darwin, if there was a ref, would be quite perturbed at somebody having published the theory beforehand, and there created a fair amount of competition. And also the colour red, the link between baboons and lipstick was a bit problematic. But we really did enjoy it. Let's have some time for a few questions. Anybody want to kick off? You've stunned them all, I think, into silence. John.
Well, I'm absolutely delighted about the comment in relation to colour red. We studied colour vision, and I was quite concerned that as a result of less evolutionary pressures, over the next 500,000 years, we're going to lose our color vision, but now I'm reassured that we're still going to have good red colors.
Yes. Well, it's also possible that the theory that, of course, I think came from this university was that women actually have 4 types of colour receptors because, of course, on their X chromosome they could have a duplication of 2, which means that instead of just having 3 colour receptors like us poor males, they could have 4 and therefore could actually see much, much better, particularly into the blues, which is why when I wear my black suit and I'm told, no, it's the navy blue suit, and I say, you mean the black one? That explains it to me, because I now do know there are some humans that can see more into the blue than I can, and they're all female.
That's extremely helpful to those of us. There's a question over there.
What do you think is next in the evolution of the eye?
It's happening all the time as we look around us. You evolve an eye that you need for the environment you live in. So what would happen if I was a flatworm living in a fairly toxic stream that is full of mercury, and I needed to move out of there fairly quickly, perhaps into some open water? I'd develop a lens fairly smartly. And I've got the toolbox to do that, because that toolbox existed 580 million years ago, and it's called PAX. PAX6, PAX-type genes, whichever ones you like. So I'd evolve a lens, and I'd do that very, very smartly. It doesn't have to be a sophisticated lens. It's just got to be enough now I'm in the open water, and I can't hide under the muddy stones and leaves in the thing. And then after that, I would probably, if that environment also became unpleasant, toxic, or I found was too full of things like trout and salmon wanting to eat me, I'd probably evolve some extra optics and do something in the dark to be out of the way of them. And I'd probably get a large cornea to do that. And I'd probably make the eye a little bit bigger. And I'd probably invest a little bit in visual processing and perhaps develop a bit of the brain. If you're talking about what humans might do, I think we'll probably develop another cone at some stage, accidentally. I don't think it'll give any survival advantage, and I think it will die out. And I think that could well have happened several times already without us knowing about it. We do know that there are people developing cones that have no function at all and leave them with just 2 colour vision, or even 1 sort of colour channel to compare. So there are numbers of evolutionary things happening in eyes, but they're not necessarily... I'm trying to avoid that term, as you notice, called evolution of the fittest, because it was kind of stolen from Darwin, if you like. But essentially, let's use that term. It's not going to make that progeny any more fit to survive in our world. Now, our world is only for a limited time, maybe 3-4 generations. This won't look like this anymore. It might look something like a medieval Holocaust. And maybe in 5 generations' time, it might look something that's really, really, really toxic, with lots of awful things happening. And different sort of vision will be needed. Maybe we will become, as I've said, nocturnal again, or maybe there'll be so much dust and ash that we're searching around for the small remnants of food that are still growing or still living under the canopy of the... And if that, I'm living under the remnants of a post-industrial city, yeah, I'll have different vision than I do now, or I'll be dead.
At that point, we're having those really exciting predictions, which fortunately none of us will be able to experience or indeed
That's
study.
the problem with evolution, isn't it? Yes.
In fact, I think most of us are probably delighted. But Will, thank you for a really enjoyable, interesting and multifaceted lecture. It really has informed us enormously. Many thanks.
Thank you very much.